No, starting the broadcast. Starting the broadcast.
Kiki Justin goes. Rah, rah, rah.
Started the broadcast. Starting the broadcast. I think we probably have already started it.
We started the broadcast.
Mutiny.
I think we started the broadcast.
Maybe you should start the show. I mean, it's 8:05 hours. Maybe we should start it.
Yeah, start the broadcast.
I like our new intro theme.
This is TWIS. This Week in Science, episode number 635, recorded on Wednesday, September 6th, 2017. Fire and flood. Hey everyone, I'm Dr. Kiki, and tonight on This Week in Science, we are going to fill your heads with Zika, black holes, and sneezes. Go first.
Disclaimer. There comes a point when it's time to get out, when the fire alarm goes off, when the forest is full of smoke, when the ship is sinking, it's time to get out. When the floodwaters are rising, when a hurricane is headed in your direction, when downtown is 10 feet underwater, it's time to get out. And for those who have doubted the predictions of global warming, who doubted the data, the scientists, and the studies, who have bunkered down in denial, it is time to get out. We have seen heroic efforts to rescue neighbors, evacuate neighborhoods in Texas and Florida. We see the destroyed homes and billions of dollars in damages. And this is just the beginning of what climate change has in store. And while no expense will be spared to save lives, on the day a hurricane hits, we could spend much less and do much more to protect those lives simply by listening to scientists. Speaking of listening to scientists, it's time to get out This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We weren't flooded out. We weren't fired out. We are still here, and we have a show to do. The show of science that is constantly ongoing. So much science news this week. I have news stories about finding black holes, CRISPR contention, and dogs. What'd you bring, Justin?
I've got ancient feet, old Americans, real vampires, and cultural wife swapping.
Whoa. All right. Blair?
Yes.
Animal. Wait, what?
So happy to be back this week. I have a story about how cleanliness is next to sexiness. I have a story about an update in bird navigation. And I have a study about radioactive sushi.
My favorite kind. How do you order that again?
What? How do you order radioactive sushi?
Yeah.
Well,
Yeah,
you put it through a conductor, and then you just it glows, I don't know.
Luminescent.
I don't know. All right, everybody. Well, let's jump into the show. Let's get a move on so that we can talk about luminescent unagi and more. But first, I want to remind everyone that you can subscribe to the TWIS podcast on iTunes, in the Google Play podcast portal, Stitcher, Spreaker, TuneIn. You can find us on YouTube and Facebook by searching for This Week in Science. It is so easy. Or just visit twis.org for show notes and audio episodes galore. But right
Sometimes
now,
I can't tell if you're saying random meaningless words
Yeah.
or if those are actually places that people
Yes.
can find us. Like
Yes.
there's so many places that we can be found now that I don't recognize them. Even having heard them week after week, they still sound new to me.
One wonders if you're listening to this list, how are you hearing it now?
I would love to know. Yeah,
Oh,
we would
yeah.
love to know these things. Let us know. And also, just listen right now because you know what time it's for?
It's our favorite part of the show.
That's right. What has science done for me lately? Yes. Good science, Dr. Kiki, Blair, and Justin. Science does a lot for me every day, from giving me my cell phone to my thyroid medicine to better insulation in my house. But today, I'd like to give a shout out to veterinary science and medicine. My cat Henry is just over a year old, but his first year of life has been pretty eventful in a medical sense. First, he had a wicked, long-lasting, around 2 months case of conjunctivitis. His vet worked with me and helped me keep him as comfortable as possible while also preventing any secondary infections. When he finally got over that, he had several months without any problems. Yay! But then he came down with an epic case of stomatitis. Ooh, that's no good. Apparently, poor Henry's immune system was fiercely attacking his teeth. It was so bad that the vet didn't think there was any way to save any of his teeth, and all indications after the fact point to that diagnosis having been completely correct. The vet thought she'd have to do 3 or 4 surgeries to remove his teeth, but when she got in there, she discovered that his teeth were so degraded that they just came out really easily. Because of that, she got almost all of his teeth in one surgery and was able to take the rest out during the second surgery. During the surgeries, the vet did many, many x-rays, yay science, to make sure that she got every piece of every tooth out so that his body wouldn't just keep attacking whatever bits got left behind. Henry was given antibiotics, yay science, to prevent any infection, and painkillers, yay science, to keep him comfortable while he healed. Thanks to science and veterinary medicine, Henry is healing up really well. Better, in fact, than expected. He can even eat dry food now, though I make sure he has plenty of wet food since it's obviously easier. Henry is an incredibly sweet cat, and I'm glad that science and veterinary medicine have been able to help him, as well as so many other injured and ill animals. And she has given us a picture of the cat, Henry, and his sister. Let's see if I can get this up on the screen for everyone. And his sister, Bess, and they're napping together. And Bess is the Siamese, giving Henry, who is gray, a hug. And don't worry, those of you who are concerned about birds. Oh, nope, I can't get it for some reason. My URL signature. Why?
It'll be posted in the show notes.
I'll put it
or
in
on
the show
Facebook.
notes.
I'll
If
put it on
you're, if you're
Put the
interested
picture
in
on our Facebook.
Yes, if you're interested in seeing a picture of this cute kitty cat cuddling, I wish I could share it right now. It's not working. But
My no cat picture filter seems to have accidentally.
it accidentally worked. But those of you who are worried about the birds outdoors, Bess and Henry are indoor cats.
Good.
So they aren't killing any birds. And this whole letter is from Minion Joni Waldrup. Joni, thank you so much for writing in and letting us know about your cat Henry and how veterinary science has helped your cat. And you're right, so many other animals to heal in times of illness. And especially now, there are many animals who are being injured and being made ill by the forest fires, wildfires that are burning across the West, and hurricanes like Hurricane Harvey who have flooded Houston. Many animals are being helped by science at this moment in time. So thank you for that wonderful reminder and about veterinary science. And also, everyone out there, remember that we need you to write in to let us know what science has done for you lately. What does it do for you every day? Send us a Facebook message on our Facebook page, This Week in Science. That's facebook.com, This Week in Science. And we want to fill this segment of the show with something from one member of our audience every week. One member of the Minion community every single week of the year. We want to do that. Help us do that. Write us with your stories and your thoughts on science and how it helps you. And you know what? It's time to move forward into the science news.
Science news time.
You know what science does for me every week? It brings me science news.
Yay.
Makes me so happy. So my first story tonight that I am really interested in talking about is a CRISPR story.
Time for
We
This
love
Week in CRISPR.
This Week in CRISPR. But this is interesting because about a month ago or so, I reported on the CRISPR gene editing study in which an Oregon, but multinational team, but led by Oregon researchers at Oregon Health and Science University, had taken care of a genetic mutation with CRISPR, edited a single mutation that is involved in a heart disease problem. And the original story, the team led by researcher Shukrat Metalipov had many experiments in dozens of embryos to correct this mutation. That's the MYBPC3 gene mutation. And they replaced this mutant that was carried by sperm with a normal copy, or so they say, from the egg cell, and were able to give an embryo with two normal copies of the gene so that there was no hypertrophic cardiomyopathy that would have been spread or given to the embryonic individual. Anyway, they also found they introduced a healthy version of the gene with the CRISPR machinery as well, but then found that the embryos really liked this maternal version better. And so they're like, okay, we put the CRISPR in there with the sperm and it's somehow this is working. They were placing their understanding of how it worked on the CRISPR machinery. However, a team out of Columbia University and Memorial Sloan Kettering Cancer Center in New York and Harvard Medical School in Boston, Massachusetts, have written a preprint bioarchive paper contesting the results of this paper. According to these researchers, they say that's not going to be possible to happen. It just couldn't happen. And their explanation is that this genetic mutation in the sperm being corrected based on the egg's version, that maternal version of the gene, they don't think there's any way that it could have happened during the reproductive period, during fertilization. So the issue that they raise is that the genomes in the egg cell, once fertilization happens, the genomes from the egg nucleus, right, and the sperm nucleus reside at opposite ends of the egg cell, surrounded entirely by a membrane that doesn't break down for hours. And so the researchers say, if there's this hard to pass membrane and the genomes are completely separated from each other, really, can you explain to us how CRISPR-Cas9 could get into the system to fix the sperm's mutation and take that gene from the egg and put it into the sperm? Can you just explain to us how that happened? That's basically what this paper says. The quote from reproductive biologist Anthony Perry from the University of Bath says, it's very difficult to conceive how recombination can occur between parental genomes across these huge cellular distances.
I like that very British way of saying it. It's very difficult to conceive this idea.
It's also
Metallic-
like how it's conceived and we're talking about.
Yes.
Conception.
Yes, that's true. Yes.
So Metalipov, the researcher who is involved in the original study, says the critique leveled by Egli et al. Offers no new results, but instead relies on alternative explanations of our results based on pure speculation.
Well, that's a fair point. If they really want to contest it, they should probably try to replicate it and say that they couldn't.
Yeah. So right now,
It's a good question.
This paper is contesting their results. It's in bioRxiv right now. Hopefully it's a preprint server, so it hasn't actually been peer reviewed yet. Maybe at some point they will get their commentary published, and then using the scientific publishing process that occurs, Metalipov and his team will likely be able to rebut every point that's in the paper and be able to explain how their data may suggest a mechanism. And other researchers may take this criticism of Metalipov's research and then try the experiments themselves to see what exactly is happening. So what Egli and the others who published this paper say is they think Metalipov's team they were misled that they had misled into thinking that they'd actually corrected the mutation, but because of their genetic assay and what genetic assays they were using. And they think their genetic assay wasn't really able to detect a more likely outcome, which is that CRISPR actually just deleted the paternal gene, and then that only picked up the fact that there was only the maternal gene in there. So they think that maybe Metalipov and his team actually think they got something when in fact they didn't. And so now what it's going to do is say, this is basically going to ask them, Metalipov et al., to come and say, okay, show us your assay works. And so now they're science in action. Are we fixing genetic mutations? We don't know yet. Is CRISPR really working the way that they think it is? We don't know. Now it's a little in question.
So it's not just that. So at first I was thinking, well, okay, so there's an unknown mechanism, maybe it worked, right? But if they can't necessarily trust their result, then yeah, they just knocked out the gene that they were going to go back and look for, and it's not there because they knocked it out, which would seem too straightforward and simple.
Yeah.
Yeah.
So there are points raised in this paper critiquing the original study that should be rebutted and approached by Metalipov's team, so that everyone knows that the work they did actually stands as they say it does.
I really hope everybody's wrong. I hope the result is correct, and but the mechanism by which it happened was like it didn't happen the way the team doing the experiment thought it did.
So you could say
but
that
also actually
wrong.
Did it, which everybody can be wrong and progress science. At the
everybody's
same time.
right, or everybody's right,
or everyone's right. Wait, what?
Yeah. The researchers did a successful experiment; they fixed it, but also the contesters were correct in it being incorrect.
And it didn't happen the way you said. Yeah, there you go.
Yeah,
both right, both wrong.
Your pipette's half
doesn't
full.
matter with science.
As I see it, pipette half full.
That's
right. It really kind of doesn't matter when you're doing science like this, and you're on cutting edge of science, and you have something that you can hold in your hand or in your data and call a result. You now have the puzzle of figuring out how you got that result exactly, and that's regardless of it's what you thought you were going to get or what you wanted to get, because you shouldn't have even wanted to get anything. You should have tried really hard not to want something, even though you suspect something is going to happen. You should really push that to the peripheral and get your result, and then figure out how you got that result. And I love the fact that this conversation is brewing right now.
Yeah,
this is fun.
Yeah, and we've seen this happen with big scientific stories throughout the time that we've been broadcasting, and it's always fun to watch the scientific conversation and see how it plays out. Who's going to be right? Who's going to, who's going to be wrong? How did they do it? Did they, how is this all going to play out? That's what I want to know. And so we will be reporting on this, and the next step will be to find out what Metalopov and his team come back with.
And it's also always interesting to I always think of Woo-Suk Wang, who did the cloning of a dog, the South Korean scientist, who it turned out the way he did it was either bad record keeping or just straight out fabricating how he got to the result. Like there was this whole controversy about this. The methods that he showed couldn't be reproduced anywhere, and there were problems. He kind of fell from grace somehow in this for some side stuff going on, but those dogs were cloned, and he's gone on to clone other. So there's also like, wait a sec, he got the result, but may not exactly have known how, right? And that might have been part of the problem. He may have tried to show proof after the fact or show process after the fact that wasn't exact, and that's why he couldn't be reproduced. But yeah. That's the wonderful thing about having an open scientific community is you get to see what's going on in the other labs.
And it's all influential and important because it moves the science forward. Even if it's wrong and you put something out there that you thought was right and it's wrong, people check it and and then it moves you back and then you move forward because it's constantly adding new information about how things work.
And if the only reason we went to the moon is because Earthlings really were hoping it was made out of cheese, we still went to the moon.
That's right. We did go to the moon. And you know what? We also are looking at maybe, that we should stick Zika virus in people's brains on purpose.
Yikes.
That sounds like mad science. That sounds like some sort of...
I know. Okay, I'm moving on to the next story.
Dark evil conspiracy theory.
Yeah, moving on to the next story. So in this dark evil conspiracy theory, it's actually taking advantage of properties of Zika virus that researchers have noticed by looking at the way it attacks the progenitor cells to neurons in the developing brain, and maybe thinking that Zika could be the tool that we can use to treat glioblastoma, which is an incredibly deadly form of brain cancer. People who get it die within a year. Usually it's we use chemotherapy now, but the chemotherapy we use gets rid of the tumor, but not the cancerous stem cells that are left in the brain. And so usually the glioblastomas make a comeback. They come back again and again, and eventually people succumb to the glioblastoma. So the cool thing here that these researchers at Washington University School of Medicine in St. Louis have been doing with researchers in conjunction with researchers from the University of California, San Diego School of Medicine is infecting mice with injecting mice with glioblastomal cells in their brains with this Zika virus. And they found that it seems to work. So they tested whether the virus could kill stem cells in glioblastomas that had been removed from patients at diagnosis. They infected tumors with one of two strains of Zika virus. Both of the strains infected and killed the cancer stem cells. They don't kill the tumor cells. All right, they kill the stem cells for the tumor, but not the tumor cells themselves.
So it's going to be a 1-2. You still got the chemo.
Exactly.
But then this is like the cleanup.
Yeah, so the researchers actually say that this is the way they're seeing it. Eventually one day being used in combination with other therapies that eradicate the tumor, but this will get rid of the stem cells potentially. And so then they checked into mice, right? They injected Zika virus or salt water as a placebo into the brain tumors of 18 mice and 15 mice, respectively. So 18 of them got Zika virus and 15 mice got this placebo of salt water. And 2 weeks after injection, they found that in the Zika-treated mice, tumors were smaller, and the mice lived longer than the mice that had gotten the placebo. So what I was going to go on to say now, it's still Zika virus. And so the question is, you stick Zika virus in the brain, you have dividing cells and progenitor cells still in your brain as an adult. And you don't want Zika virus going after all the cells in your brain and potentially setting up an infection that could, you don't want to have Zika in your brain, right? So now the next thing that they want to do is to create mutant Zika viruses that could be safe to use as just a vector that use the mechanism and the machinery that Zika uses, but then doesn't set up an infection. And that's
so you know how sometimes one of the arguments against deforestation in the Amazon or South America, what have you, is that we may be destroying plants that hold cures for diseases, and so we should preserve them, if nothing else, for the future of medicine on this planet. There may be plants undiscovered there's something to be said about diseases now, because we're starting
right, yeah.
CAR T, the one that they used as they're using as leukemia cure, is using a mutated HIV.
Absolutely,
virus.
Yeah. We use the lentivirus, absolutely, and we are.
So we're like, wait. Now we just forget malaria, like curing malaria disease or getting rid of the mosquitoes. We got to figure out where we can use malaria to cure disease. I mean, one of the wonderful things about these pathogens that overcome our body's natural defenses and attack cells is that we're at a point where we can redirect them and have them attack the cells that we want them to attack. Well, we're getting there. We're finding ways.
We're learning about it. Yeah, and they also teach us how our immune system works.
Yeah, and how when it fails, we might be able to overcome those failures. So these pathogens, I'm glad we've got, as odd as it sounds, like we have a CDC lab with a this influenza or that horrible disease, or maybe Ebola. We're gonna be like, I need to get an Ebola shot this week. I'm really, whatever the disease is, we may be getting to the point where we can co-opt and hack these things that overcome our immunity, our immune system, to do good for the body, to attack something that's a worse threat and disarm it at the same time. Then when they're doing the CAR T for leukemia, patients aren't ending up with HIV as a result. They're getting cured of their leukemia. If they can, define which aspects or which portions of the Zika virus are the ones that they need to do the targeting and attacking, they can probably make a mutated version that doesn't have the other aspects of it that aren't specifically needed. So you need to, of course, understand the full mechanism of how this works.
We do, yeah. And that we are not near that yet, but these do offer opportunities to learn a lot more and potentially, like you said, create great ways to treat our illnesses. And
potentially in ways. There's always all these caveats when we're talking about medical cures coming forward.
Yes, because this is years and years and years away. It's in mice right now. They haven't figured out how to make it safe for people. I'm not going to let you run down the train of no holds barred here, Justin. I mean,
no, that's not where I was going. That's not where I was going.
But we need to it in a little bit and stick with this is where the science is right. Eventually it'll be Lego bricks of biology.
What I was saying was, though, look how amazingly quick those turnarounds are now. From a discovering of a way to do these mutations to a potential cure. Whereas when we first started doing this show a long time ago, years, talking about maybe lead to open a door towards a pathway to research into, we were talking 20, 30-year time frames. Now we're talking about they discovered this and there's trials underway, and this within the next 5 years, 4 years might be a treatment that's available. CAR T took less than that.
Yeah, CAR T. No, yeah, I
CAR
think
T didn't take that long.
10 to 15 years. I'm gonna keep it. I'm gonna temper your madness there.
Rein him in, Kiki. Good work.
Yeah, but you know what?
The turnarounds are quicker.
You know what was also coming quicker?
What?
Human natural selection for longevity.
Great.
That's
Live
right.
forever.
Yeah. Researchers publishing in PLOS Biology analyzed DNA from 215,000 people to look for parts of the genome that might be in the process of evolving. So I looked at US and UK databases for mutations whose prevalence changed across age groups. So as ages got older or younger, did the proportions of these mutations change? Because that would suggest that they're being selected for some aspect of survival, especially if they say increase or decrease with aging. Researcher Hakamanesh Mostafavi, who's at Columbia University, said if a genetic variant influences survival, its frequency should change with the age of the surviving individuals. So if you have a harmful mutation or allele for a gene, you're probably going to die more, right? Bigger rate of death. And so if the population at a whole is dying more because of this genetic variant, then that genetic variant becomes less prevalent in the old part of the population, right? So they looked at more than 8 million common mutations, and they only found two that seemed two that became less prevalent with age. Only two. One was a variant of the ApoE gene, which has been linked to Alzheimer's disease. They really didn't find it in women over 70. Another mutation was found in the CHRNA3 gene, CHRNA3 gene, which is associated with heavy smoking in men, and it starts to become less around middle age. So what they say is that if people don't have these mutations, they're more likely to survive and live longer. And so the authors argue that this large study and the fact that they found only two variants, two genes, really suggests that evolution and natural selection is weeding out lots of harmful mutations, and that there are many others that are probably already gone.
But aren't we now, with all of our cutting edge medical research abilities or application abilities, going the opposite direction? I mean, what if actually all the increases in the Western world of diabetes or this disease or that disease actually is because we can treat everything now? We can treat so many things that they don't leave the gene pool. So it's not so much that the disease is increasing because of diets or environment or any of those things, but just because more people are alive who can have the genes that support these diseases.
That's interesting.
That's an interesting idea. Yeah, but there's
pure speculation, but yeah.
Yeah, that is speculation for sure. Yeah, but if we could be supporting harmful mutations that would have been weeded out. But the assumption that underlies the study still stands, which is that if they're harmful, deleterious to survival, then
but are they counting diabetes in that, or is that like no longer considered harmful? So there's a whole bunch of factors that go into that. But you can also say that part of our immunity evolution is science. I mean, that's part of how we've evolved, is we've added science to the toolkit. So, yeah, I guess their central point then.
Yeah, that wasn't their central point, but it's an interesting point to bring up. Yeah, there are other things like fertility and longevity, and this idea that one of the ideas that they think is that genes related to longevity or that are harmful towards longevity could potentially be less harmful and not really, that we just maybe haven't really noticed how harmful they are, or that they're connected to decreased fertility as well. And so maybe there's something tying together also decreased fertility and decreased lifespan that really reduces the prevalence of mutations. But there are lots of interesting questions here to bring up about people and where we're going. And I love the idea that, you know, regardless, we're surviving, and there are fewer of these deleterious mutations that are kind of kicking around, and that maybe the longevity genes are the ones that are really going to be sticking with us. That's why everyone's getting older. I don't know. Other research that came along with this also, I mean, not with this study, but United States, fatherhood has officially passed the 30-year-old barrier. The average age of fathers in the United States at first child is over 30 at this point. An aging population, older fathers having children. Maybe this increases longevity genes.
I think it probably does.
Yeah.
Young fathers probably do a much worse job at parenting.
Here's some fireworks. Oh, sorry. All right, this is This Week in Science. Justin, what you got?
The history of humanity is not entirely our own. We know that that many humans or human-like creatures called hominins walked the earth at some point. At some point, we all shared a common ancestor. And as we crossed paths, we crossed genes and shared other more recent common histories more closely with each other. But there are far more tales of the tailless ape than we have the knowledge to tell. Case in point, new footprints from Western Crete. They have an unmistakably human-like form, especially the toes. The big toe is similar to our own in shape and size. It also has the distinct ball, that big part in the back of our foot on the sole, on our sole of our foot, which is never present in apes. They don't have that ball in the sole. So the shape indicates that these footprints that they found in Western Crete belong to an early hominin, and they were made on a sandy seashore, possibly in a small river delta. What makes these footprints most intriguing, though, is not that they were found, but how old they are and where they were found, as the footprints are approximately 5.7 million years old.
Whoa.
Huh?
That's old.
What? Very extremely old. And in fact, at a time when previous research puts our ancestors in Africa and with ape-like feet, not these
human-like
clearly
feet.
More toed human feet.
Yeah.
Human feet have a very distinctive shape. They're actually, we are different from all the other animals in our feet. A combination of a long sole, 5 short forward-pointing toes, no claws for most of us, and a big toe that is larger than the other toes. That is unique. That's a human trait. Not totally modern human trait. It's a little bit older human trait to ancestral human trait, but it's a human thing. Feet of our other close relatives, the other great apes, they look more like a hand with a thumb that sticks out to the side, right? They're not as footy. So there's an iconic footprint, the Litoli footprints from Tanzania. Those are 3.7 million years old, and they show human-like feet and upright locomotion. Those are the oldest footprints of such feet until this one, which is 2 million years older.
That's crazy.
Not in Africa. The Litoli feet are thought to have been made by Australopithecus. They're actually kind of look a little bit more like the modern human foot, those 3.7 million year old feet. And the heel, except that the heel's a little narrower than ours, the sole lacks proper arch. So you go back before this Litoli Australopithecus, go back into the fossil record another 700,000 years. you will find the 4.4 million year old Adropithecus ramidus from Ethiopia. This is the oldest known hominin from a reasonably complete fossils, and it has an ape-like foot. Ape-like foot, and then you got to go back another 1.3 million years to get to Western Crete and find a more human-looking foot. Adropithecus ramidus was considered ancestor of Australopithecus. So now, maybe not so much. Possibility, Australopithecus, much older, or whatever made these footprints. The Western Crete feet is not as long as the Australopithecus latoli. They I guess are a little shorter. I'm not guessing. It says they're a little shorter, a bit more primitive looking, perhaps. At approximately 5.7 million years, they are younger than the oldest known fossil hominin. That's in Chad, but those were ape-like feet back then.
Wow. So, okay, I think the big thing here is it's the timing. I mean, when were these footprints made? Can we really prove 5.6 million years ago?
Marine
the
fossil
location,
record, below and above. Below and above is how they did the dating, and that's where it's sitting. So it's also interesting.
This is crazy.
I guess they point out this earlier this year, another group of researchers reinterpreted fragmentary 7.2 million year old primate that was Graceopithecus that was found in Greece and Bulgaria. They redefined or reinterpreted this ape, even though it's only known from teeth and jaws. They're saying, you know, these look hominin. These don't look primate. These look like they could have been hominin at 7.2 million years. So the story of our history is still being filled in. And now part of why this is revolutionary sounding is because we were at some point all trained, told, thought, and all of the research in this field said you went from having ape feet to human-like feet. It was a straight line progression. Now we have the braided stream theory that tells us that you had ape feet running around the same time you had human-y feet, and you had several human-y feet and plenty of ape-like feet all on the planet at the same time. So it should be expected that Australopithecus could be on, walking about making footprints the same time ape-like feet are still present in Africa. What it changes most dramatically is our perception of how long humans have been wandering about. And if it turns out hominins have been wandering about. And it alters a little bit of history that we already knew wasn't entirely the way that things happen. So we've gotten enough clues lately not to be too surprised by this, but still 5.6 million, 5.7 million year old footprints in Greece of all places that are looking very Australopithecus like, or kind of a primitive shorter version. So maybe the ancestor of or an early version of Australopithecus. That's really going to be fascinating. So yeah.
Very.
Yeah.
I want to hear more about this as it progresses.
Yeah.
Scientific conversations.
Yeah.
Changing our understanding of human evolution.
A bit, and another little human history type story. Analysis of the skeleton found in Chan Hole, a cave near Tulum, Mexico, suggests human settlement in the Americas occurred in the late Pleistocene era. So we've had plenty of evidence. This is the 13,000-year-old bones that have been dated. These are human bones, skeleton they found in this cave, dated to 13,000 years ago. Not at all surprising. We have shell mounds, we have structures, we have all this other evidence, some of it even a little bit older than this. But this is the oldest dated skeletal remains in the Americas found in this cave in Mexico. So not an alteration of timeline, but another pin in there that illustrates that, like the shell mounds, like some of these other structures and fragments and tools that we found, the skeleton is representing another pin in that map, saying yes, we are pretty well drilling down the colonization of America, and to be in Mexico 13,000 years ago, and perhaps as long as 25,000 years ago, when you get up above into the Canadian sort of Alaskan tundras. People's been around America for a really long time.
Yeah, I mean, stuff like being able to date fossils like this is it's important, because we used to think that man barely had appeared on the North American continent 13,000 years ago.
Mm.
You know,
it
was
depends
like,
on when
it used to be, oh, it was like what, 10,000 years ago, 13,000 years ago. We were fighting about that. And now it's like going further and further back. And now it's 13,000 years ago.
It's further back than
Mexico.
The current estimation is like getting to North America was at least, if not more than, at least 20,000 years ago. But there was all these glaciers in the way. So then, finding folks and structures 13,000 years, that was a big. I think when I was in school, I think it was getting updated from like 7 to 9,000 years.
Exactly. That's what I was. That's what I was kind of referring to.
Yeah.
The date keeps going further and further back, and actually being able to do this skeleton based. I mean, this is a skeleton. Yeah. That's an individual who died in a place, right? An isotopic analysis, being able to date it and say 13,000 years ago ish.
Yeah.
That's
and then it's sort of. Yeah. I guess let's see, where's it? They examined human skeletal remains found in the cave. Researchers dated the skeleton by analyzing uranium, carbon, and oxygen isotopes found in its bones and in the stalagmite, which had grown through its pelvic bone. It's been here for a while. Been here for quite some time.
Yeah.
You've got a stalagmite through your pelvic bone.
You're gonna have a bad time.
You haven't moved in a really long
while.
time.
That's right. Well, is it time for some moving and shaking of This Week in Science? Yeah. Because you know what time it is now?
What time is it?
It's time for Blair's Animal Corner.
She loves our creatures, cried and small. Biped, millipede, no pet at all. Wanna hear about animals? She's your girl. Except for giant pandas and squirrels.
What you got, Blair?
Oh my goodness, I'm so excited to be back. I have a really exciting story about a very special bird called a golden-collared manakin. Golden-collared manakins, the males will form a lek. That's a group of males that display together to attract the attention of females. They all kind of gather into one arena, and then the females get to come and take their pick. The way the lek works for these manakins is they have a display area on the forest floor that they protect, that they defend, and they clean. And then the females pick their favorite. So, a recent study was specifically looking, this is the Smithsonian Tropical Research Institute in Panama. They had a very kind of unique opportunity because of the Smithsonian Institute in Panama to study these animals in the wild and do some slight tweaking to wild birds. So they basically were able to combine an observational study in the wild with lab conditions, because this is such a controlled space in the Smithsonian Tropical Research Institute. So what they wanted to see was if hormones influenced the male mannequins drive to clean up. The way they did that is they treated some young males with testosterone and then watched their movements in the display court. They also removed they removed some leaves, they added some leaves, and they weighed down some of the leaves to make them heavier to see how hard they were really willing to try to clean up their space. And what they found was that testosterone-treated young males got super into cleaning.
So these male birds on roids
Yes.
had a little cleaning roid rage.
Yes, absolutely. They removed significantly more leaves than untreated males. They even pulled up small seedlings.
Wow.
And they were so motivated to clean up that they flew carrying leaves, weighing as much as their own body.
That's like that's like you're picking up another person. Like,
Yes, that's correct.
It's just like the human feat of picking up a car.
Beyond that, the younger males were more innovative in the ways that they cleaned their space. So basically, if a leaf is really heavy, they're going to have to try a bunch of different kind of plans of attack to get the space clear of the riffraff and the clutter. So what they saw in this study was that hormones are playing a role in motivating birds to court mates because the cleanliness is directly related to females picking them. So the more testosterone, the more likely they were clean. They were going to clean their space more kind of vigorously. In order to kind of test this hypothesis across the board, they also treated some females with testosterone. When they did that, females were not motivated to clean, but they did become more aggressive and show male display behaviors to other males. So they were affected by the testosterone, but this is where it got interesting, is they didn't clean. So what this is telling the researchers at the Smithsonian Institute and UCLA that partnered for this study is that hormones alone are not what causes the cleaning behavior. The testosterone is acting on neural circuits and things in place that indicate to them. The testosterone tells them, I'm anthropomorphizing a little bit, but they're so excited. They really want to win over a lady. They know that a clean space will win the lady. Upping the testosterone ups the overall effort. But because females don't court, the testosterone just made them really aggressive, but it didn't make them clean.
Right. So they started acting like other aggressive males,
Right.
but not in the trying to woo the males or in trying to woo other females. They didn't they didn't go toward those male behaviors. Yeah. Interesting.
Looking around my place, I'm glad that not all females are looking for a clean forest floor.
Justin, they all are. Moving on from cleanliness to smell. I guess they're kind of related. I want to talk about birds in another area of study, and that's navigation. We've talked a whole bunch on this show. I feel like every few months I have a new update in the world of bird navigation, but that's kind of what makes it so interesting, is that when understanding bird brains and understanding how birds get around, we still only have pieces of the picture. And Kiki and I kind of went off on this crazy tangent a few months ago about bird smell. For a very, very, very long time, people thought birds don't smell. But then what happened, Kiki?
Oh, those silly
Birds can
researchers realized that they'd just been cutting off the olfactory bulb and had been not considering the fact that birds could smell because they didn't think they had the brain organ for it.
You silly researchers, you pulled the brain pan off and off went the
olfactory bulb, all those
olfactory
neurons.
bulb. So it turns out this whole time, birds can smell. This is in the 90s, early 2000s even, a lot of the world thought Birds can't smell. Turns out they can. So there's this whole new world of research now looking at bird smell and how bird smell affects their behavior. Well, enter bird smell in navigation. This is so fascinating. So a combined effort with Oxford, Barcelona, and Pisa, researchers from all three of these places have worked together to look at olfaction with bird navigation. And they found that navigation, they particularly wanted to look at the hardest place for birds to navigate, which is over open ocean with no view of land. How do birds know where they're going? So in previous experiments looking at this, they actually displaced the birds. They would take the birds, they'd move them to a new spot and see if they could find their way back. And then when they did that, they would do sensory manipulation. They would change their magnetic or olfactory receptors. They would do some sort of deprivation. From these experiments, they had suggested that removing a bird's sense of smell impairs homing, but disruption of magnetic sense, they couldn't find any conclusive results. These researchers wanted to try something completely new, and instead, they wanted to deprive these birds of certain receptors or abilities to sense certain things, but not displace them. Just plop them back where they came from and see how they acted in a normal environment, which to me, again, I'm not in the intricacies of bird research, but it makes more sense because you're not tweaking as many variables at one time. So what they did is they looked at 32 scopuli shearwaters, and they split them into 3 groups. One, they made temporarily anosmic, so they were unable to smell through nasal.
You can't smell.
Right. Through nasal irrigation with zinc sulfate temporarily. Another, they had them carry small magnets, so their magnetic sense was all messed up, and then a control group. And then they attached GPS loggers to each of them. And they did this while the birds were nesting, so they had no reason to leave the space. The birds were incubating eggs in crevices and caves on the Menorcan coast. So this way, they could grab the birds, they could mess with them, put them back. They were fairly certain the birds would go right back to their eggs because they wouldn't want to abandon them. And that worked. So then they tracked them as they engaged in normal foraging trips. While they're sitting on the eggs, they have to leave periodically to feed. The birds went out on their trips as normal. They gained weight. Everything seemed normal. They returned and exchanged. They did incubation shifts with their partners. So removing a sense of smell and messing with their magnetic senses did not impair their motivation to return home or their ability to forage. But what it did do is when they made birds anosmic, when they took away their sense of smell, they made successful trips for foraging, but their orientation on wide open sea was odd. So they went straight. They didn't go all over the place, but they didn't go in the right direction. And then once they could see land, they redirected and went straight home. So they were close, but they weren't going exactly where they're supposed to be going. So the implication here, the idea is that when they don't have visual cues, they use olfaction as a factor in their navigation. So
That would make sense.
it would. You would think an animal would use all of their senses when
And they're
doing
not-
things.
And the interesting thing to this is that it brings up is that the magnetic sense, even though the birds have a magnetic sense, what they're doing is they're foraging. They're not migrating.
Right.
So these birds are not instinctually driven to use the magnetic sense to be able to get them from one place to another directionally. Like, because what they've done is they've gone out over open ocean and they're like, I'm just looking for food.
Right.
Which way did I go? Oh, now I just need to follow my way back.
Right. I think about if you were going to the grocery store versus if you were driving out of state. You might
Yes.
turn on your GPS if you're going somewhere far away you haven't been before, or it's been a long time since you went there. But if it's a place you go every day, you don't need your GPS. However, if you usually go during the day and then it's night and one of your landmarks you're used to seeing, oh no, it's not there anymore, you might get a little less certain. You might make a couple wrong turns, but you'd probably make your way back home.
Yep.
So
It's kind of similar potentially to the way that salmon use their olfaction scent to get them to their right home stream,
yes,
get them home. Get
absolutely.
me home. Smell it home all the way. That's interesting. I love it. Love it. Ties in nicely with other past stories related to this.
Absolutely.
Love it.
The saga
Bird
continues.
nose. Yes.
Yeah.
And birds get their sense of smell. It's so amazing. We find out more and more about it. This is This Week in Science. It's time for us to take a break. We'll be back in just a few moments. With more science, we've got dogs and black holes and some other things coming up in just a few. So stay tuned.
Can't explain the things you heard with more than intuition. The line of reason shows the way.
Hey everyone, thanks so much for watching or listening to This Week in Science this week. We do appreciate that you are here with us once again to talk about all the science that we want to talk about, and bird noses. Bird noses are great. You know what's also great? Pre-ordering the This Week in Science calendar. You know, it is available now. This Week in Science, you can go to twis.org, which is the wonderful portal to all things TWIS. Go to twis.org. And right now there is a link up on our website for you to be able to pre-order the 2018 This Week in Science Blair's Animal Corner calendar. And this calendar will be getting mailed out in November, December time. We're hoping to sell out, so we're making the ordering earlier this year, so you can get it on your list of items to get for gifts, for just for yourself, for a calendar for next year. It'll have all sorts of wonderful science holidays in it, days that you should be aware of. The TWIS schedule, at least as far as we know it, for 2018. But right now, you can go to twis.org and pre-order that calendar. So head on over there. Additionally, while you're over at twis.org, if you want to check out our Zazzle store, click on that Zazzle store link. It'll take you right over to zazzle.com/thisweekinscience, where we have all of the other wonderful This Week in Science products. And there are other things that are Blair's Animal Cornery related. There are mouse pads and phone covers and lumbar pillows that are all from past Blair's Animal Corner This Week in Science calendars. Additionally, if you are interested in figuring out how you can help support This Week in Science, one easy way is to donate to TWIS. You just click on the donate button. That allows you to go through a PayPal platform, payment platform, where you can pay the amount that you decide you would like to at that particular time. Or you can click on our Patreon link, which is in the main header bar. The Patreon link will take you over to our Patreon community, patreon.com/thisweekinscience, where people support the show in an ongoing way from episode to episode and month to month to really keep this show going and keep it on the interwebs and on the air. You can pledge however much you decide you would like, and we give you little gifts in return. I will mail them to you through the mail, not the interwebs, but the mail. I will. It's a lot of fun stuff there. This Week in Science, where you can find all of our past episodes. Click and listen to the latest episode. Also see the show notes and links to stories There's all sorts of stuff at twist.org for you, and where you can investigate how you can help support This Week in Science and help produce it in an ongoing way. If another thing that would be super helpful is if you could help share TWIS. Tell people about twist.org. Tell people about how to subscribe to TWIS on iTunes, how to subscribe to us on YouTube, how to find us on Facebook, all these places where we live. Help people find us. That would be wonderful. We'd love more people to find us. We thank you for your support. We really couldn't do this without you.
Displays. Now due to the juices and pills and the creams, your body's lost toxins, whatever that means. You've stopped eating all of that sinister food. Your dinner tastes awful, so it's gotta be good.
And we're back with more This Week in Science.
Yes, we are. Justin, what you got?
Oh, what did I bring? Aha! Is it possible that a mythical creature once used to frighten children before putting them to bed could actually have been real?
Nessie?
Nessie.
Yeah, it could be Nessie. Bigfoot?
Sasquatch?
What about vampires?
Oh,
I always tell my kids about vampires right before putting them to bed.
sure.
Father of the year, ladies and gentlemen.
Vampires have a lot of mythical roles to fill these days. Nowadays they come out in the daytime. They sparkle. They're glittery. There's all sorts of versions of this. But the classic, the pale-skinned, blood-drinking nocturnal creature who's harmed by being in daylight may actually describe a very rare but very real blood disorder. Erythropoietic protoporphyria, also known as
Erythropoietic protoporphyria.
yeah,
What she said.
aka EPP, which I will call it EPP from here on out. It affects the body's molecular machinery for making heme, which is the component of the oxygen transporting protein hemoglobin. When heme binds with iron, it gives blood that red look. EPP causes people's skin to become very sensitive to light. Prolonged exposure to sunshine can cause painful, disfiguring blisters. Quotey voice, people with EPP are chronically anemic, which makes them feel very tired and look very pale with increased photosensitivity because they can't come out in the daylight, says
So does
Barry
living in
Pa,
Portland.
PhD.
Oh.
Yeah, actually, even on a cloudy day, there's enough ultraviolet light to cause blistering and disfigurement to those exposed body parts.
Terrible.
So staying indoors during the day and receiving blood transfusions containing sufficient heme levels can help alleviate some of the disorder's symptoms. In ancient times, this may have been achieved by drinking animal blood and emerging only at night. Yeah.
Fascinating.
Paz and his team of investigators report in a paper in Proceedings of the National Academy of Sciences the newly discovered genetic mutation that triggers EPP has been discovered. It illuminates a novel biological mechanism potentially responsible for the stories of vampires, seeing as how the EPP folks kind of have this, some of the symptoms and express some of the things that we've come to associate with the creatures of the night.
Neat.
Yeah, you go outside, it burns. Yeah. What
Do
a
people have
terrible
this
disorder.
now? Yeah, do people have this now? Is this still a thing?
Yeah, it's a
Yes.
thing. But I mean, that's what they're figuring out, like potential cures for, and they, in the or at least the genetic causes of. But yeah, it's a real thing.
Yeah.
Terrible. I feel very
Fascinating and sad,
fascinating and sad. Exactly.
but also fascinating.
And maybe someday these ideas of vampires will be just a thing of the very, very long past. Yeah. Yeah. Interesting. Other creatures of the night. Well, how about not really the night, but maybe the dark universe around us? Black holes, right? We always try to see them, but we don't really see them. The only way we really see them is kind of by the way they move gas around and the effects they have on the space around them. Basically, we have to infer that black holes are there because, black holes, they absorb all the light, so you can't really see them. So we have to infer where they exist. Really, really supermassive black holes, pretty easy to infer at the center of galaxies. You've got these black holes, jets of material coming out of the area where the black holes exist. Little tiny mini black holes, also fairly easy for us to find. We have been able to discover a lot of them. However, the most difficult time we have had is in finding mid-sized, intermediate-sized black holes. So these mid-sized black holes, if we find them in the centers of galaxies, we've got a big giant one, a giant supermassive black hole in the center of the Milky Way. But if there's another more mid-sized black hole hanging out in the Milky Way somewhere, that could indicate that the Milky Way grew and got bigger by cannibalism and by sucking up its neighbors, and that maybe it's in the process, that supermassive black hole, Sagittarius 1A, that it's in the process of sucking this other black hole into it still. And so researchers have been kind of looking for this, and they've finally found evidence. A team led by Tomohiro Ukua of Keio University in Yokohama, Japan, found a very peculiar cloud of molecular gas called CO-0.40-0.22. And this cloud of molecular gas was close to the center of our Milky Way. It moved with a large range of velocities, some of them very, very, very fast, which made them think that there's really something quite massive in there that they hadn't seen yet that's influencing the velocity of the gas. And they simulated the movements that they saw, and their simulations said this might be a black hole of about 100,000 solar masses. Wow. So they went and they took the Atacama Large Millimeter Submillimeter Array, otherwise known as ALMA, to observe shorter wavelengths from the radio frequency spectrum to see very fine detail in this molecular cloud of gas. And they found a really dense clump of gas near the center of the cloud. And this clump in the center, it was even more fine-grained in the resolution of velocities that suggested a really massive nearby object. And so then they simulated it. And again, the simulations were very supportive of a massive black hole. So the spectrum of the source was really similar to the supermassive black hole at the center of the Milky Way, but much less luminant. About 500 times less luminous. And so they think, the researchers say, that this supports the notion that this molecular cloud, this radio source that they found, is an intermediate mass black hole, and that it may support this idea that the Milky Way cannibalized a neighbor at one point in time, because this black hole that they think they're seeing is just too big to have just gotten a start there in our Milky Way. It doesn't make sense based on our models of galaxy formation. There are about 50 dwarf galaxies near the Milky Way, and it would support the idea that galaxies like the Milky Way grow through cannibalism if this molecular cloud is confirmed to be a black hole.
I just love that sentence. Black holes grow through cannibalism.
That's right. Cannibalizing. The super suckers
Well, that's
of the universe.
that's quite. I mean, that's what they are, right? They're a whole bunch of material that's.
Yep,
mushed
they're giant
down.
suckers. Yep, you got it.
Yeah, so it's like
the only way that they get.
So like spiral galaxies like ours, most of them probably have a big supermassive black hole at the center, but maybe, if they formed in a little galactic nursery at some point, there was nest cannibalism at the galactic scale.
Too much. Little baby black holes. Not the babies.
Sucker. Moving on up and into closer proximity to us, our solar system, Juno has been out hanging out, taking a look at Jupiter, and has been sending back some really amazing information about the gas and the atmosphere, the depths of this planet. One of the big things that's come out recently is new information about how auroras form.
Is that Biz on top? Is that what that is?
Yeah, so I'm showing a picture right now from NASA that was taken by Juno of an aurora lighting up the northern pole of the planet. And it's a wonderful bright blue color illuminating the top of the planet. Here on Earth, we have aurora that form when electrons in our high atmosphere get sped up, and because of the magnetic fields that are propelled around our planet, and those magnetic fields push those electrons around and basically shove them into gas particles, and energy is released in the form of light. And so at our poles, we see aurorae, I guess, from the North and the South Pole. However, this news out of Jupiter suggests that we thought that Jupiter had, or that Jupiter's auroras were caused by the same process as here on Earth. But what Juno is showing us is that's not the case. Well, it is some of the time. Some of the time it is. There's a lot of plasma in the atmosphere, and there are electrons in the outer casing of Jupiter. And Jupiter does have a massive magnetic field. And so, yes, of course, there are aurora and electrons that emit light in the way that Earth's upper atmosphere does. But that's not the predominant mechanism for how aurora happen. Really, what it's more like is that there are these waves in the planet's magnetic field. There's a lot of turbulence in the magnetic field because it's so massive, and there is a lot of plasma and electrons and things that are in that upper atmosphere that get pushed around, just pushed like a wave. And so there's a lighting process that this wonderful article from Science analogizes. It's a process somewhat akin to surfers being driven shoreward ahead of breaking ocean waves. So if you can imagine these turbulent waves pushing the electrons forward and energizing them, it's more as if the waves are breaking and energizing the electrons themselves, as opposed to the process of knocking the electrons like billiard balls into gas particles in the atmosphere, which is what happens here on Earth. So it's a completely different process. Thank you, Juno. We never would have known without you.
Neat.
Yeah. Anyway, this is it's going to help us understand more about what's happening in the atmosphere, Juno, and potentially, hopefully, understand the magnetic field of Jupiter a lot more, which would be awesome. What else you got, Justin?
Let's see. Oh yeah. This is the medieval wife swapping culture. So this is end of the Stone Age, getting into the early Bronze Age. Families were established in a surprising manner in the Lecto community, which wasn't much of a community. It's sort of just a fertile ridgeline area where there was farms and stuff. This is in south of Augsburg, Germany, if you're familiar with the area. Based on the remains of 84 individuals buried between 2500 and 1650 BC in cemeteries that belonged to individual homesteads, they found the majority of women came from outside the area, probably from Bohemia or central Germany, while the men buried there, usually remained in that region from birth to death. This is a so-called example, or an example of so-called patrilocal pattern, and it persisted over a period of 800 years during the transition from Neolithic to early Bronze Age. Cody voice, we see a great diversity of different female lineages, which would occur if over time many women relocated to this Lech Valley from somewhere else. This is, remarks Alyssa Mitnick on the genetic analysis. And Karina Knipper explains, based on analysis of strontium isotope ratios and molars, which allows us to draw conclusions about the origin of people, we were able to ascertain that the majority of women did not originate from the region. Whether these women were traveling of their own free will or somehow being kidnapped, cursed, or sold into matrimony isn't clear, though once living the farm life, it seems they integrated just fine. At least the burials of women did not differ from that of the native population, indicating that the formerly foreign women were part of the local community by death. Researchers suspect it played a significant role in the exchange of cultural objects and ideas, which increased considerably in the Bronze Age, in turn promoting the development of new technologies. And if the pattern was expanded out to the greater region, the exchange of culture and ideas that drove this age would have been the women who were moving from one place to another and bringing with them the traditions or practices or knowledge of where they grew up and coming to this Lech Valley. Also, it could be the origin of the word lech.
No, it's so interesting. We have these different periods of time culturally when, we had British monarchy, royalty, who were inbreeding because they just married the other rich, the landowners, right? And then we had this other period of time previous to that where it was like, no, we're traveling. The men were traveling. They would find women, bring them back, and start their families with someone from far away. Fascinating how things change. Fascinating. Always changing. Another thing that is going to change, hopefully, maybe, is our concept of dog intelligence. We love dogs, right? Dogs, man's best friend.
Dogs are great.
Dogs are great. But until now, dogs have never passed the mirror test. Now, what's the mirror test? The mirror test is a test of self-recognition and thus self-awareness and metacognition and consciousness. It gives a lot of ideas as to how thoughtful an organism is. So if you take a dolphin, for instance, and you put a mark on its side, that dolphin will swim around in front of a mirror, checking out that mark on its side. It spends a lot more time in front of the mirror if there's a mark on its side than if there isn't a mark on its side. People, children, if you put something on them babies after a certain age of development, if you put something on them, they will spend more time looking in the mirror at themselves.
Yeah, we did this. We did a story about them doing this with monkeys, where the monkeys, instead of reaching for the thing in the mirror, they reach for it on themselves first.
Yes.
Yeah.
Yes. So that shows that there's self-awareness and self-recognition, which is a sign of higher intelligence. People who own dogs, people say, yeah, my dog's smart. My dog knows me. My dog knows itself. The dog knows it. But until now, scientists have never been able to show this awareness. But you know what? We talk all the time, Blair, in the Animal Corner and about other animal behavior studies about appropriate tests to ask the right question. Dogs are not primarily visual. They are primarily
Sensual.
They are primarily olfactory in nature. They spend the first thing a dog does is not go look at another dog, they'll go sniff another dog. A dog spends a lot of time in this odor world. And so, what researchers did is they designed an analogous test to mirror self recognition, in which they took dogs' own scent that from the dog's own urine. And then they gave another stimulus in which the odor, the urine scent was modified somehow. And then another scent, which was a completely different dog's scent. And they found that dogs can actually distinguish between this olfactory, what they say, quote, image of themselves when modified. They investigated their own odor for longer when it had an additional odor accompanying it than when it did not. And they think this implies a recognition of the odor as being of or from themselves. And so the ecological validity of this was examined when they also presented the subjects with other dogs' odors, right? Unknown dogs or known dogs. And dogs spend longer investigating the odor of other dogs than their own odor. And then in the last experiment, they spent longer with the modified stimulus than with the modified odor by itself. So that novelty is not the thing that explains the behavior.
It makes sense. I mean, with the monkey study that we did a couple of months ago, it was all about teaching the monkeys how a mirror worked, and then they immediately passed the mirror test. So this is the problem. There aren't mirrors in the middle of the wild. So testing based on a mirror, you might as well test an animal based on a Macintosh.
Right.
It's a level of technology they don't understand. So this is really cool. I love it. I think the more we can adjust our methodology of testing intelligence of animals, the better.
Yep.
Absolutely.
Yeah. So dogs, self-aware, they have self-recognition. They can distinguish their own odor from that of other dogs. They know when their odor has been altered. Mm-hmm. I think it's a very well done study of this question of whether dogs have self-recognition, and then the idea that they do. That's confirmation. Whew. All right. You know what other dogs though, wild dogs, African wild dogs. This story blows my mind. Researchers, oh my goodness, this was academics from Swansea, Australia, the United States. They were looking at these endangered African wild dogs in the Botswana Predator Conservation Trust. They found that these animals use sneezes for a social decision-making purpose. They used the sneezes as quorum sensing to decide when they were going to move off to hunt after making camp for greeting ceremonies in what they call social rallies. They used to think that the dogs were just sneezing, clearing their airways. All these dogs were together, sneezing.
I'm allergic to dogs. Oh, me too. Oh, so am I. Oh my goodness.
Yeah.
But when the researchers looked at the details of 68 of these social rallies where the dogs came together, they found that when there were more sneezes, it was more likely that the pack moved away to go hunting. And so they've decided that it's quorum sensing, and sneezes are the way that the animals vote and decide to do this together.
I love the story. It also said that the vote or the sneeze of the alpha male or the alpha female was more important. It took less sneezes after that for them to move.
Yeah.
So, but here's the thing about this.
Sneeze is more powerful than yours.
We can't call it a sneeze anymore. It's not a sneeze. This is not a reflexatory movement that was done in response to dust in their nose. This is a vocalization that sounds like a sneeze, right?
Yes.
So I don't know if they want to call it an outward chuff or what, but after I read this story, it took me a while to process it because it seemed so ridiculous, and I figured out that's why.
Because they're calling it a sneeze.
And I kept thinking about it like a sneeze. And why are they sneezing on commit? No, it's a vocalization. It's just a different sound that they make. But it's fascinating that they in their lexicon, they've developed this one very distinctive sound, unlike any other that they make. That is a vote to move on.
To move on. Vote with your sneezes or your outward respiratory chuff.
Yes. There we go. See, fixed it. Hashtag fixed it.
Hashtag fixed it. My final story for the night is another communication story, this time from the bird world. We've talked before about the cuckoo.
Oh, the brood parasite.
Brood parasitic common cuckoos, Cuculus canorus. They lay their eggs in the nests of a host species, and the female will often move around and figure out where appropriate hosts' nests are and pay visits very secretly to the nests to drop the eggs in. And it's only been known, however, that the males of the cuckoo species do a lot of vocalization, and they have a very a call that is used during the breeding season. And in the experiments that these researchers did that are published in Nature, Ecology, and Evolution, they found that the reed warbler really doesn't pay any attention to the cuckoo call of the male cuckoo. They don't, any other bird, the cuckoo, they don't pay any attention to it. However, the female cuckoo makes a weird chuckle call after it lays its egg in a host's nest. And they realized, the researchers discovered, that this chuckle call is actually copying the sound of a hawk. It's a hawk-like call that the warblers actually spend more time looking out of the nest, away from the nest, when that chuckle call is made, and it diverts their attention. So that the parasite's egg is just like, oh, there it is. That must be mine now. I got distracted. Oops.
Hawk! Squirrel! Hawk! Another hawk! Squirrel! Squirrel! Hawk!
You're jimmying the lock, and you're saying your car alarm's going off.
Exactly.
Jimmying the lock to the house. Don't look at your front door. Go look at your car through the window.
Wow.
Yep. So the female cuckoo is enhancing her success by manipulating the host's concern about being prey.
Oh, the cuckoo, the ultimate con man.
Ultimately. Now, Blair, you have a story.
Oh, I have one last story. The headline, ladies and gentlemen, everyone, can you all just calm down? Calm down.
I can't
calm
down.
About Fukushima and the fish. Let's calm down. Just everyone settle. Simmer down now. So a recent study actually looked very carefully at radioactive isotopes in the Pacific. They really wanted to put this to bed. Okay. So an international research team showed that concerns about radioactivity in fish hurting humans, we can put it to bed. It's not a concern. They looked at marine predators like tuna, swordfish, and sharks. You look at predators because of biomagnification. If there's even a tiny bit in the ocean, in these large predators, it grows exponentially up the food chain. So the predators will show, they're the smoking gun. They will show if there's really anything to worry about in the ocean. So they focused on cesium, which is a silvery metal. It has a lot of radioactive isotopes. They looked at 134C and 137C, which is a form that is found when uranium fuel breaks down in nuclear reactors. So they're what is discharged in large quantities following a radioactive disaster. That's what you would look for that would tell you that there's something wrong with the fish because of Fukushima. This stuff, it also has relatively long half-lives, 2.1 and 30 years. So considering that it wasn't that long ago since the Fukushima disaster, they should be able to find it. And it tends to accumulate in muscle tissues, which is what we're eating. So this is really what they wanted to look at. And they sampled across the Pacific between 2012 and 2015. This was a big study. They found no detectable levels of 134C and 137C, concentrations that were generally consistent with background levels from above ground nuclear testings in 1940s and 1950s were that's all they found. What they found was stuff that had been there since the 1940s and 1950s. They did not find anything higher than that.
Wow. So even since the nuclear testing period, basically these nuclear disasters, Fukushima melting down, it hasn't been anywhere close to the amount of stuff that we put into the environment when we were doing nuclear testing.
Yeah. So what they found, for marketed fish to be restricted from trade, it would have to have been 1,600 times higher than what they measured. So one of the co-authors, Kevin Wang, he says, go ahead and eat some sushi. So go ahead and eat some sushi, everybody. It's kind of the moral here is, and I hesitate to say this, but it's true, the ocean's really big. And so this idea that this radioactive disaster is going to affect fish that are washing up on our shores here in California or Oregon or Washington, it's not a concern. The things that we should be worried about in our waterways are other things, like the effects of climate change, like the effects of ocean acidification, like the effects of the 300 million pounds of Atlantic salmon that were released into Pacific waterways. Whoops. So those are the things to worry about. So go ahead, eat your sushi. Don't worry about the Fukushima thing. When it comes to that, when I say go eat your sushi, though, still be smart. Pick safe fish. Make sure that the restaurants you're going to have good quality sushi grade fish. And if you're pregnant, obviously you don't want to eat too much sushi. But don't worry about the nuclear activity. That's what I'm telling you here today.
Fantastic. I wasn't worried about it, but for those of our listeners who might have been, I hope that this assuages their fears. And we'll have a link to the story in our show notes when the show notes are all put together so that you can check out the story yourself if you want to find out more and have a place to start your investigations.
Yeah, I know we've talked about it a lot on the show, but I still know lots of people that are saying, oh, I'm cutting down on the sushi, that Fukushima stuff. I know people who are usually fairly scientific who are saying these things because it is widespread in a lot of the media and the social media. People are still very up in arms about it. So
Really? Wow.
I just want to send out some real science about this issue.
Thank you. Yes. Much appreciated. Yeah. Really? Ah!
Yeah, I know.
So it's fish, like at all, like they're eating.
They didn't find anything higher than the 1940s and 1950s testing.
No, what I'm saying is like the people who are like, yeah, I'll go down and have fish and chips because that's British. But,
yes.
But sushi.
They're worried about the raw. I know people specifically who don't want to eat the raw fish.
Roast,
which I do find hilarious because cooking it is
not going to do anything.
Radioactive isotopes.
No.
Just cook out the uranium. It'll be fine.
I have heard a lot of people say, I'll eat the Atlantic fish. I'm not eating the Pacific fish right now. That is something that I have heard pretty widely as well. So here's the actual science. Read it at will.
Careful,
There
because
you
your
go.
Atlantic salmon probably came from the North Pacific.
Right. There you go.
Oh, you're gonna lose no matter what. And whitefish, you have no idea what kind of fish that
whitefish
is. Or
might as well be eating fish.
no way to
All right.
Just a white meated fish. That's it.
Did we
Oh my
do
goodness.
it?
We have done it. We have
we've
come
done
to
it.
the end of another episode of This Week in Science. So much
Great.
science this week. What a fun show. Everyone, I would like to start our shout outs for thanking everyone for their support on Patreon. Thank you to Paul Disney, G. Burton Lattimore, John Ratnaswamy, Richard Onum, Byron Lee, E.O., Kevin Parachan, Jacqueline Boyster, Tyron Fong, Andy Groh, Keith Corsell, Jake Jones, Gerald Shirelles, Chris Clark, Richard Hendricks, Charlene Henry, Brian Hedrick, John Gridley, Steve Bickle, Kevin Railsback, Ulysses Adkins, Dave Friedel, James Randall, Bug Calder, Mark Mazzaros, Edward Dyer, Trainer 84, Layla Marshall Clark, Larry Garcia, Randy Mazuka, Tony Steele, Gerald Onyago, Steve DeBell, Louis Smith, The Harden Family, IFSHMN, Greg Guthman, Patrick Cohn, Ksenia Volkova, Daryl Rinsrang, Alex Wilson, Jason Schneiderman, Dave Neighbor, Jason Josier, Matthew Litwin, Eric Knapp, Jason Roberts, Richard Porter, Rodney, David Widley, Robert Aston, Sir Fricadelic, Christopher Rapin, Dana Pearson, Paul Stanton, David Brennan Binish, Dale Bryant, Todd Northcutt, Arlene Moss, Bill Curthy, where did I end that? I missed one. Bill Kersey, Ben Rothig, Darwin Hannon, Rudy Garcia, Felix Alvarez, Brian Hone, Orly Radio, Brian Condren, Mark, Nathan Greco, Hexator, Mitch Neves, Flying Out, John Crocker, Christopher Dreyer, Artyom Shiwata, David Wilskin, Steve Mishisky, Ramus, Gary Swinsberg, Bill Nadeau, Braxton Howard, Sal Good, Sam, Matt Sutter, Emma Grenier, Philip Shane, James Dobson, Kurt Larson, Stefan Insama, Honey Moss, Mountain Sloth, Jim DePoe, Jason Olds, James Paul West, Alec Doty, Lumala, Joe Wheeler, Dougal Campbell, Craig Porter, Adam Mishkan, Aaron Luthen, Marjorie David Simmerly, Tyler Harrison, Colombo Ahmed. Thank you for all of your support on Patreon. And for those of you who would like to support us you can find information at patreon.com/thisweekinscience. Also, remember, you can help us out simply by telling your friends about TWIS. That is a super help. Share TWIS with your friends. Really, really great help. On next week's show, we will be back once again with more science news, and we'll be broadcasting live online at 8 p.m. Pacific time on twist.org/live, where you can watch and join our chat room. But don't worry if you can't make it. Our episodes can be found at twist.org/YouTube, at facebook.com/thisweekinscience, or just twist.org.
Thank you for enjoying the show. TWIS is also available as a podcast. You can simply Google This Week in Science in your iTunes directory. Or if you have one of those mobile type devices, you can look for TWIS, the number 4 Droid app in the Android marketplace, or simply This Week in Science in anything Apple marketplacey.
For more information on anything you've heard here today, show notes will be available on our website. That's at www.twist.org, where you can also make comments and start conversations with the hosts and other listeners.
Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in your subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there is a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to in the night, please let us know.
We will be back right here next week. And we hope you'll join us again for more great science news.
And if you learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science.
This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
This is the end of the after show. Blair looks concerned.
Just reading a thing.
Reading a thing. Gong. We have made it to the end of another show. I'm going to tell everybody now I'm not. Hi, Panoramic. Hello. Hello, Ben Rothig. I put you guys up on the chat room for a second. On the screen. Put the chat room up there for just a quick second. Justin said he had a story to tell, but now he's not here. And I am not going to stay very late tonight. I really need to go to bed because I have an early morning tomorrow.
Fair.
Which is fine, but I got to tell this story first.
I would love to hear it.
Okay.
Yes.
This is it's not really story like a sciencey news story exactly, but it follows along the path of an explorer. Let me pull this up. Okay. So this is out of Australian National University. An anthropologist at the Australian National University may have stumbled across a clue to resolving one of the most enduring mysteries of Pacific history, the fate of famous French navigator Jean-François de Galloup, Comte de la Pérouse, who disappeared in 1788. La Pérouse was instructed by King Louis the XV, 14th?
Yep. The Sun King.
to undertake a major voyage of exploration in the Pacific to emulate the feats of Captain James Cook. I guess he was a little jealous. He departed the French port of Brest in 1785 with 2 frigates and a complement of 225 officers, sailors, and scientists. This is Dr. Garrick Hitchcock, the ANU School of Culture, History, and Languages. He believes the last survivors of Le Pursue's voyage were shipwrecked on the Great Barrier Reef near Murray Island in northeast Torres Strait. La Perouse's voyage of discovery in the Pacific is recognized as one of the most important of its era. Rivaled only by the work of Cook, he remains a very well-known and respected figure in 18th century scientific exploration, Dr. Hitchcock says. What is known is that La Perouse's ships Astrolabe and Boussole were wrecked in 1788 on Vanikoro, a small island in the Santa Cruz group of the Solomon Islands. The survivors made it to shore, spent several months constructing 2 small masted craft using timber salvaged from the wreck of the Astrolabe. Once completed, they launched the vessel in a bid to return to France. What became of this ship and its crew, desperate to return to France, has been an ongoing mystery. While researching a project on the history of Torres Strait, Dr. Hitchcock came across an article published in 1818 in an Indian newspaper, the Madras Courier. He is confident the article reveals what became of the survivors. The article tells the story of Shaikh Jumal, a castaway Indian seaman who survived the sinking of a merchant ship, Morning Star, when it was wrecked off the coast of North Queensland in 1814. Jamal made it to Murray Island, where he lived for 4 years, learning the language and culture of the islanders, because you can't really call an Uber back then. Shipwrecked on an island. Okay, I'm home. Hi, my name is Jamal. What's yours? He was finally rescued by 2 merchant ships that passed through the area in 1818. Jamal informed his rescuers that he had seen cutlasses and muskets on the island, which he recognized as not being of English make, because, of course, being Indian, from India, that's what he would have recognized, as well as a compass and a gold watch, he said, were on the island. When he asked the islanders where they were obtained, they related how approximately 30 years earlier, a ship had been wrecked on the Great Barrier Reef to the east in sight of the island. Boats with crew had come ashore, but fighting followed. All were eventually killed, except a boy who was saved and brought up as one of their own, later marrying a local woman. The Perceau expedition crew list includes a ship's boy, Francois Mordelle, from the port of Tigre in Brittany, northwestern France. Dr. Hitchcock wonders if Mordelle could be the last survivor of this expedition. The Indian newspaper article featuring the castaway's account was later reproduced in several other newspapers and periodicals of the day in Australia, Britain, France, and other countries, and observers noted that this might refer to that specific expedition, says Hitchcock. Somehow, Sheikh Jamal's story was subsequently forgotten. While a French book published in 2012 refers briefly to this newspaper article and discounts it as an unreliable account, Dr. Hitchcock believes otherwise. The chronology is spot on, for it was 30 years earlier, in late 1788 or early 1789, that the Le Pursuit survivors left Vanikoro on their small vessels. Furthermore, historians and maritime archaeologists are not aware of any other European ship being in that region at that time. This means that this is the earliest known shipwreck in the Torres Strait, and indeed Eastern Australia. Could well be that the final phase of the La Perouse expedition ended in tragedy in Northern Australia. Further recovery of artifacts from the wreck site on the Great Barrier Reef, yet to be discovered, or other islands, will hopefully provide the final confirmation. This particular Torres Strait, which includes the northern part of Australia's Great Barrier Reef, is studded with reefs, rocks, and sandbars, and has been described as a graveyard of ships. Over 120 vessels are known to have come to grief in its treacherous waters. Mm-hmm. I thought that was a fun story. A secondhand account that sort of that fits the timeline And the secondhand account is, another castaway's observation of, there's a lot of technology and artifacts that don't really seem to belong here. Isn't that odd? Oh, there's a story that goes with it. I'll recount that. And then, 100 years later, plus, we're, starting to figure out.
It's funny.
Oh, yeah. Maybe we might know exactly whose junk that was, whose artifacts,
Too
whose
late
stuff.
for him, but oh well.
Specifically whose.
Yeah. Yep. Nice.
But
And with that,
was a fun story.
Kiki, you can cast away into
Cast away.
I was just looking at
Castaways.
I'm going to Lexington, Kentucky tomorrow.
Lexington, Kentucky.
Yes, it is the horse capital of the world. Turns out.
Yes, it is.
Yes, indeed.
Yes, horse capital of the world.
Horse.
Lexington, Kentucky. Second largest city in Kentucky. Horse capital of the world. Heart of the state's bluegrass region. Yes. This is interesting. I went to one of my favorite things, Atlas Obscura. I wanted to see like what unusual stuff there is in Lexington. And they have a medical and science museum with life-size figure casts from as many as 200 cadavers.
Whoa.
Outstanding collection of 19th century medical instruments. Yeah. Pretty interesting. I'm gonna have to go there.
Yeah, do it. How long are you there? For the weekend?
Yeah.
Conference?
No, my best friend from high school just moved there, and she's flying me out to help her pick out a dog.
Interesting.
Yes.
Panoramic, who is from Australia and in Australia, points out from the previous story there that James Cook got eaten. So I wouldn't want to emulate him.
Oh man, this museum's by appointment only. Atlas Obscura, what are you doing? There's a floral clock. There's the grave of Daniel Boone. In Frankfurt.
Fada in the chat room.
Wasn't there a song about Daniel Boone?
Well, first off, most fish or sushi that is eaten on the west coast of the U.S. is not from Japan. Wouldn't be very fresh. Yes. Just the idea that it's Japanese food, I guess. It's like, oh, no teriyaki bowls either. No teriyaki
It's more
chicken bowls.
fresh on the western U.S. than it is on the eastern U.S.
Yeah, because we actually have sushi fish that
comes from Japan. Like,
yes.
Yeah,
but also a lot of it you can get on the Pacific,
West Coast,
on the west coast of the U.S.
because of where they swim.
Yeah, because it's,
and
you
that's
know,
why
it's
people were all
one
worried.
big ocean.
One big
So
ocean. One big ocean. Although there was a restaurant, I remember, I haven't been there, but I know people who say it's the most amazing sushi. Tiny restaurant in Hayes Valley for sushi. And I guess they fly the fish in fresh from Japan every morning.
Okay. Did you know that in Lexington, Kentucky, there's a water tower the shape of a Dixie cup?
I did not know that.
There's also a pharmacy built in the shape of a mortar and pestle. And there's a biblical golf course.
Here it is.
And on the 7th hole,
Yes.
the golfers rested. There's a nice bench and a picnic table.
Oh my god.
Sort of have lunch on the 7th.
I found it.
Hole of rest.
Oh my goodness.
Well, have fun.
Boone was a man.
Thanks.
Was a big man.
I'll try to not get in trouble.
And his stick is as mighty as an oak tree. There's the Daniel Boone theme song. You don't remember that song?
No.
I shared it in the chat room.
So my friend told me also to keep an eye out at the Lexington airport because it's teeny tiny for horse airplanes.
Oh, that would make sense.
So
Yes,
she was
horse racing is
saying
big over there.
the horse capital of the world. So I've heard.
Yes.
What she said was, in order for them to be like real legit race horses, they cannot be a product of insemination. It has to be natural breeding. So that means that they
makes a difference.
Yeah. So they fly horses across the country to mate with other horses to make thoroughbred horses.
Boy, doesn't that make you think that if like somebody wanted like somebody a little bit science minded might be able to just come up with a better racehorse tomorrow.
Yeah.
Like, you know, like what age are they pulling their horse racing knowledge from that they're sticking to?
Yeah, I don't know.
in this day and age?
But yeah, she was saying you can tell them because there's no windows on those airplanes.
Yeah, because that would scare a horse.
Well, I'm sure they're drugged.
Well, it might scare a drugged horse, and who wants that? There's gonna be a hoof flying out a window. Horse is like, man. Yeah, that could be. It could just be bad.
Eric in Alaska says the Denver airport has a museum dedicated to the conspiracy theories about the Denver airport. I've been to the
That's
Denver
awesome.
airport. I do not recall that.
It's a big airport. You'd have to gone everywhere.
Well, we're going again.
And
soon,
besides,
right?
that
So
museum
let's
is in
do
the
it.
basement, and you need secret deep state knowledge to get clearance to go down.
Well, I know that like when the giant horse
On the
statue
40th
there,
floor.
when it flashes the red
42nd floor.
eyes, that's a signal to the Illuminati. But
Mhm.
yeah.
Signal to the yes. No.
Okay, I'm now making fart
No.
sounds with my mouth.
Okay.
What?
Good night,
Probably
everybody.
come to the end of the usable content.
I'm trying to figure. Yeah, I think we've gotten to the end of it. I'm going to try and find somebody to talk to about the Cassini mission next week.
Oh, cool.
I think on the 15th it makes its final dive into Saturn.
Yeah, my prediction, it pops out the other side. And we're like, what?
No, it's going to get
That's
crushed.
really my prediction. No, it's instead of getting crushed, it just pops out the other side. And we're like, oh boy, there's really nothing there. Huh?
We will all be crushed. We will all be crushed by the end of Cassini. That is my prediction.
And on
Right,
that cheering
hit
note,
return.
good night, everybody.
Yeah, no, is there anything else? I don't think there's anything else that I need to talk about that I'm going to get the... Yeah, nope, we're good for the weekend. We're good for the week. I hope everyone gets to high ground if you're in Florida. Stay safe if you are in... Please stay safe. Don't listen to Rush Limbaugh.
Huh?
What's he doing?
Are they really saying don't worry about it?
No. Yeah, Rush, did you hear? Rush Limbaugh said terrible things.
What did he
Wait.
say? That's not a big deal?
Yeah, he said don't listen to the liberal media, that they're saying it's a big, that Irma is a big deal when it's really not, and that
That's terrible, man.
that you don't need to go to the store to buy any supplies because it's all a conspiracy between the liberal media. I'm paraphrasing, of course, but it's a conspiracy between the liberal media and marketers and people to make people spend money.
Oh, absolutely. You know what? Absolutely. This is exactly what you expect from people who begin down the road of deciding to distrust science. It becomes this thing where they just that now it's all has to be fake.
Don't
Everything
worry.
about science has to be fake. Even
If he was there,
climatologists
if he was in a space where there was the hurricane, you better believe he'd take care of himself.
Right, exactly.
Where is he? Isn't he in Florida?
He's in LA, isn't he?
Yeah.
I
Or
don't
LA.
No idea.
Yeah, it's all over the news. You can find stories about this on the Washington Post. You can find stories about it on Fox News. Yeah.
What a jerk.
Yes.
Well,
Al Roker is calling out Rush Limbaugh about this.
The governor, what's Governor Scott? of Florida is a high profile climate denier. You know, as were senators in Texas. So the leadership that doesn't trust the data that's predicting what they're experiencing. What can you say? Yeah, panoramic. Rush Limbaugh was actually a radio host in Sacramento. Although when he was in Sacramento, believe it or not, he was a liberal voice on the radio.
No.
Yeah.
He is. He's
he followed the money.
Opportunistic.
For sure.
He's just an opportunistic. He has no ideology, as I believe a lot of people with extreme ideologies don't actually have ideology behind their words, but have agendas. And
so
that
it turns out, but this is an interesting thing. We're talking about agenda, though. It turns out, and WI guy has pointed out that Rush Limbaugh is in Florida, and I just looked it up. His house is in Palm Beach, Florida, which is potentially one of the areas that could be very devastated.
Should live broadcast from there.
That'll be very
live broadcast. We're going to be streaming live from my house. We're going to be streaming live. We're gonna ride out this liberal media. I'm doing the other guy. Who am I doing? I'm doing Alex Jones. More
That's right. Do it somebody else. Yeah, interesting. But it's interesting comments. And my hope is that nobody listens to comments such as these and really just, consider the worst possible thing that could happen and prepare for that. Be prepared for the worst and be happy and surprised when it's not that bad.
And
I hope
also,
that everyone's safe over the week to come.
Yeah, and I also hope there's an investigation into the skyrocketing airline ticket fares to leave. You guys heard about this?
No.
This is the story I heard. A father who's got a daughter that's going to school in Miami, I think it is, decided just to get her back up to Connecticut and spent, I think, $160 on a ticket. Went and looked it up and got a ticket, got her a flight out. And then it turned out her roommate, which is also a family friend, hadn't arranged to leave. So the next day he went to go buy her a ticket on the same airline, same kind of plane, same flight plan. And it was over $1,000.
Oh.
So, and he looked elsewhere and found other airlines were likewise similarly.
God, can we not capitalize on this problem?
Yeah, I mean, how?
Well, that's our economy.
I mean,
capitalizing.
Yeah.
Yes.
There should be rules against this kind of stuff, though. This is a natural disaster.
Well, but this makes it so that only those with the most money can leave the terrible disaster situation.
Yeah.
And so like, there are actually one of the only laws, even though Texas or Houston didn't have any zoning laws, which allowed people to build houses or developers to build houses in floodplains. They do have an anti-emergency situation gouging law, which is pretty intense. Like, it's like $20,000 per offense. So like if you start charging $20 a gallon for gas, I don't know if it's every person that fills up or every gallon of gas, but they levy insane fines at you. Like it's so I don't know that Florida has any such laws on the book. It's going to be a state by state thing. And then but there is something really treacherous about that. The one airline that hadn't jacked up their price, from what I was reading, was JetBlue. Except that JetBlue's only flights out of the area also like did this little jump down towards the eye of the hurricane. Like they went like they had this. You got picked up here, but then you had to go here and then
That's
here.
What I was going to say. JetBlue doesn't go many places.
It doesn't, and so its route wasn't direct, and it went towards the hurricane, and so might not even be flying. Like. So yeah, I hope there's some sort of backlash.
Yeah,
there. That's awful base behavior. Just double your flights and charge half. Come on, people.
Yeah.
Standing room only. It's an emergency.
Yeah. Yeah.
Yikes.
All right, you guys. I'm wishing everyone the best in the week to come, and I hope to see you all again next week
Yes,
with more This Week in Science.
definitely.
Good night, Kiki.
Good night,
Wait, that's
Kiki. Good night, Blair. Good night, Justin.
Okay, that's a quick out. Good night, everybody.
Good night,
All at
everyone.
once at the same time, Indians included. We're saying good night to you too.
Good night.